Welcome everybody, I'm Professor Steve Forsythe and I'm going to talk today about misidentifications of Cronobacter, subtitle Hindsight and Oversights. And the reason for doing this is to encourage researchers when they look at papers to look at the date of the paper, see what the methods are based on and how things may have changed since those techniques were originally designed. Because I believe this does have a direct influence on potentially protecting neonatal health. So what I'm doing, this is like the 5th YouTube video in a series. I already did one back in March on the isolates from Michigan. I've done one on the Cronobacter sakazaki pathovar, the meningitic pathovar CC4, and also more recently I did a two-parter going through how a neonatal intensive care unit outbreak has been investigated with different techniques, progressing through being able to differentiate the strains more and more over a 10-year period. And there are a couple of links here for part 1 and part 2. The reason for this presentation is there is still a lot of issues in the press with regard to the situation in America, but primarily this is a follow-on from an ELSI and IAFP webinar that we did last week on Cronobacter in the Spotlight, New Insights into a Known Organism. And I thought, really I ought to get one out on identification procedures to try and get everybody together so you're aware of the literature. So it's primarily to help those who are not familiar with Cronobacter, who are now learning about the organism, just to help you understand the background literature. I found it very awkward to categorize the material for this presentation. And I've also varied with regard to, do I go through a particular technique? Do I go through it chronologically? Do I go through it according to organism? And it's ended up a bit of everything, as you're going to see. Now, there are 3 notes I want to like disclaimers, so to speak, at the beginning. I've been gathering information, literature for many years. Various examples of misidentifications have been occurring, and I've kept meaning to write a review. And a few of my collaborators know I keep saying, I'm going to write this review sometime. They said, please do. Never had the time. However, the situation in America has really highlighted the organism again after several years of it not being in the news at all. So I decided that there could be new researchers out there, and because of the confusing literature, then I wanted to try and clarify the story and how the story has changed over time as we've learned more and more about the organism. As I was putting this together, I realized that actually I could link it back to the FAO-WHO risk assessments on microbial contamination of infant formula. That was, in truth, an afterthought. It was as I was putting the slides together, I thought, oh my goodness, this actually links back to 2004 risk assessments that I was involved with. And I think it is appropriate to do that because, in general, we are concerned about the microbial safety of powdered infant formula, not just Cronobacter. Now, again, just to clarify, not all neonatal infections are through contaminated powdered infant formula. There are cases that were due to the water that was used to rehydrate the infant formula, and even breast pumps as well. And I got various, those other YouTube videos go through sources. That's not quite so pertinent for this one, which I've been so hard to keep focused. Now, I am not speaking as an industrial microbiologist doing routine analysis where I understand you need a technique that will work, give you a result, make a decision. I am, and generic results are more sufficient. It could be like generic question, are E. coli present in the product? Fine, that's an acceptable question on industrial scale. You don't need to know, is that E. coli 0157 a foodborne pathogen? Or for that question, you don't need to drill it down, is it E. coli K1, which is a neonatal meningitic E. coli? Okay, the question is there E. coli, yes or no? So the researcher, in a sense, or the regulator wants to like drill that down. So I'm speaking as a research microbiologist after many years, decades, working on the diversity of Cronobacter and related organisms. So I think, really, if you are detecting meningitic Citrobacter, if you are detecting meningitic Cronobacter Sakazaki, then you should be aware that that is actually the equivalent of detecting neonatal meningitic E. coli K1, okay, pathovar of E. coli causing baby meningitis. I think if you are detecting Cronobacter and you know that, then you should think seriously about it. So, as I say, these have been accumulating for years. I've been meaning to write the review for years. And you might be asking the question, especially at the end of this, have I told the authors of the articles that I'm commenting on? And the answer is not anymore. I've given up trying. In the past, I've contacted corresponding authors. I've even contacted the editors of the journal. As a referee, I've refused to referee papers, said these strains are not confirmed for what they are, and I've honestly got nowhere. I've even sometimes open access comments. There's a section on open access articles where you can leave comments. I've left comments, nothing's happened. So there's been no responses. So I have stopped trying. That even includes contacting the FDA, CDC, and a well-known infant formula manufacturer, contacting the Michigan isolates. And I made that video, did those results back in March 2022. And just to clarify, those isolates in Michigan include 3 strains of the clinically important Cronobacter Sakazaki ST1 and a strain of neonatal meningitic pathovar Cronobacter Sakazaki ST4. I've heard nothing back. Therefore, I intend to keep this video open-ended. If associated authors contact me, if I've overlooked corrections, if I've overlooked or my comments are out of context, then I will add those comments to the description below in this video. And if necessary, I'll do a follow-up one. That'd be great to do a follow-up one so everybody knows what the situation is. So the answer to have I told the authors? No, not anymore. I've stopped, I've given up, but watch this space because hopefully some authors may contact me and I can clarify the situation for everyone. That'd be great. That'd be my preference. So here's a silly little saying I made up. Where you are going depends on where you're starting from. If your initial culture collection is flawed, then your detection methods will be flawed as well. And that's a key message from this presentation. Flawed phenotyping can lead to a flawed culture collection. Therefore, when you come to design your PCR probes, they can be flawed. And if the database is flawed, then lots of people can get affected. But there are also people who unfortunately seem to ignore the literature or don't keep themselves familiar with the literature adequately. So I want to point out that the effects, and really this is very much more on the research side, that what's been happening to me has affected our identification methods, and it's actually affected our understanding of the pathogenicity of the organism. And in a sense, it's time to hit the reset button, and to me, to go DNA sequence based, not PCR probes and phenotyping. Okay, speaking as a researcher, not as an industrial microbiologist. One of the outcomes, and this was not my original intent, was I believe that there is further evidence supporting the concern over the possible occurrence of the pathogens Enterobacter cloacae, Enterobacter homaceae, and Citrobacter cosseri in powdered infant formula. And that directly links back to the first 2 risk assessments of WHO FAO in 2004-2008, and there'll be a slide on that in a moment. So please bear in mind that's what has come out when I've been pulling this together. It wasn't what my original intention was. So roughly the structure of this is I'm going through a timeline of key Cronobacter events. We're going to look at those FAO-WHO risk assessments. We're then going to do the inevitable, what is the taxonomy of the Cronobacter genus? Then, because of what's going to come later, I'm going to do the origin of the multi-locus sequence typing, according to the time when we originated it. Then the middle section is going to be looking at misidentifications in the literature through phenotyping, serotyping, even through DNA sequencing, and a couple of odds and ends just the end, just to finish off, and then references, who am I, and so on and so forth. Here's a nice timeline that was put together by a collaborator, and it goes through the issues like when things started, pre-2002, coming up to 2017. And please use this as like a reference where you can see how when detection methods were developed and what the risk assessments were, they were decades ago. We didn't have so much opportunity to sequence organisms in those days. We had limited knowledge. And so please have a look at this timeline, according to your time, and just think, what techniques am I using? When were they developed? What was our knowledge at that time? Here is the categorization of bacterial pathogens in some way associated with powdered infant formula from the WHO FAO 2004. There was an addition of Enterobacter in 2008. That's the major difference. But what we did then is that we sat down and we looked at the information. What clinical evidence do we have of infections of Cronobacter and Salmonella from powdered infant formula? Is it found in powdered infant formula? What's the epidemiological evidence? And Cronobacter, then known as Enterobacteriaceae, we just call it Cronobacter for the moment, and Salmonella were the only ones where we found clear evidence of causality. Now, in that meetings, we also considered that there was a plausible link, but not at that time demonstrated, for Enterobacter cloacae, Citrobacter fundi, Citrobacter cosseri, Pantoa, Klebsiella. There was other Enterobacteriaceae, and these like gram-positives, less plausible. Now, again, I've slightly modified this. I put Enterobacter cloacae in inverted commas because, it has, to put it politely, it's a bit of a dumping ground for some organisms for when that species was being put together, because we didn't have DNA sequencing to clarify things. So Enterobacter cloacae is there in inverted commas. You'll see why as we go through. Then I've highlighted Citrobacter cosseri. Now, in those days, we were very concerned about Citrobacter cosseri because Citrobacter cosseri, almost identically to Cronobacter, causes invasive neonatal meningitis. That means it gets through the blood-brain barrier rather than swelling the meninges. It actually gets through the blood-brain barrier and causes destruction of the baby's brain. Now, you can find pictures of that. I am not putting such pictures in this presentation. They could be upsetting, and if they could upset, they will upset. So I'm not including them. But if you do look them up and you compare them with Cronobacter invasive meningitis, you'll understand why back in 2004 we were concerned about Citrobacter cosseri in infant formula as well. So here is that original summary back in 2004, almost 20 years, 18 years ago, almost 20 years ago. And these are the executive summary. These are like the recommendations from the meetings that we had. And what they say is that we ought to get internationally validated detection and molecular typing methods sorted out for Enterobacteriaceae. And we're looking at the sources, including infant formula, and other relevant organisms should be looked at. and then we should get a better understanding of ecology, taxonomy, virulence, and other characteristics of Enterobacter sakazaki and ways of reducing. Now, what I've highlighted here is like that first bit, molecular typing, then ways of flag. You have the internationally curated open access Cronobacter PubMLST database, and I've already given links to YouTube videos on that. Down here, the genus, like Shea Fanning and Nestle and others put together and changed, clarified the taxonomy of Enterobacter sakazaki. So the whole genus comes from that work, and again you can get those in videos as well. Now, I'm the curator for the Cronobacter PubMLST database. This was like a click on the 3rd of July, so a couple of weeks ago, over 1,000 genomes, over 3,000 strains in that database, and you can submit either whole genomes or 7 loci, and there's 2 primer sets if you are doing it by PCR 7 loci. A minority of people do that these days. Majority just upload the whole genome sequence, and it takes me 5 minutes just to upload it and to assign it and everything. So matter of minutes, that's all it takes me. That gives us this picture. Yeah, this is a great picture. The Cronobacter genus, the 7 species of Cronobacter, related genera, Citrobacter, Enterobacter, Franconibacter, Cicibacter, and that looks great. But that's what we now know. When we originally, way back in time, realized that we needed to avoid phenotyping, we needed to avoid 16S for this organism, we went for multi-locus sequence typing. And these are the only 3 genomes. So this is the same picture, only now showing the only genomes that we had. Those are the 3 genomes that were used by Adam Baldwin at University of Warwick, postdoc, and he looked at loci after loci until he narrowed it down to 7 that he believed could be used, because back in 2008, 2009, that's where we were, PCR sequencing. And the only 3 genomes we had were a strain of Cronobacter sakazaki, so multi-locus sequence typing, that defined it as ST1. We had an Enterobacter sp, which had not been speciated at that time, so that became sequence type 2. And because of this invasive neonatal meningitis, fortunately, we were very keen to include Citrobacter cosseri, which is sequence type 6, in the PubMLST database. It doesn't only cover Cronobacter, it covers related species. And so we wanted to include that one because we believed at that time there could be a strong link between the 2 organisms, and the more information we had, the better it would get. So that's where we were in 2009, 3 genomes, and that's how the PubMLST database was designed, based upon those 3 originally. And that's why it's so expansive, because it didn't start with 1 species, didn't start with 1 genera, it started with 3 genera. So it's very wide, very encompassing. So let's go taxonomy, and I never was interested in taxonomy until I got into Cronobacter. It seemed to be like a black art to me, but hey ho, absolutely essential to understand taxonomy in order to reduce the risk to neonatal health from Cronobacter. Where we were by 1985 is that we had Enterobacter sakazaki. It was generally the yellow pigmented Enterobacter cloacae, as I say, is a bit of a dumping ground. And when you did your phenotyping test, there was 1 strain in the ATC culture collection, 51329, and that was your quality control strain, what's also known as a perceptual strain. So that's your quality control. Does my technique work? Does it work with this strain? Okay. So then we have 2007, the work of Shea Fanning and others, and we have the beginning of the Cronobacter genus. Bear in mind, not all Cronobacter produce a yellow pigment. Bear in mind that a large part of this was understandably based upon biotyping, when it was phenotyping. And the names in blue are the currently recognized Cronobacter species. Now that was quickly followed up when it was realized that 16S didn't reliably differentiate Sakazaki and Melanaticus. They're too conserved. Some people misunderstand. They think Melanaticus means that's the only one that metabolizes malonic acid. It's not true. There are some Cronobacter Sakazaki that do metabolize malonate. Okay, so it's not a differential test. It's a majority, but it's not 100% absolute. Then we drop that out. We jump a few years, we get the new one of Cronobacter condimenti that we did, and Universalis that again we did. And it's another story as to how we discovered and named those organisms. That's for another day. Then we have this frustrating one, 2013, where a group went and said that these Enterobacter really should be Cronobacter based upon only 5 loci. I'll skip making judgments on that for a recording. But fortunately, Lenner and Stefan, they corrected this, thank goodness, and these became the Franconibacter and the Cicibacter. Largely, there's no pathogens in this group, although there's one case for the Cicibacter tertiensis. As frustrating is that these organisms were used as negative controls, and so by putting this Cronobacter messed up some techniques. So that's a quick history of the taxonomy. Bear in mind that this information, there's a lag time for it being implemented and for it being accepted. So it's one thing to say, oh, a paper came out 2007, paper 2008, paper 2012. It takes time for manufacturers and regulators, etc., to catch up and to implement and the consequences of that. So there's a lag time which varies, unfortunately. But down the left-hand side, it's really important topics put in. The ISO 2006 that did not get replaced until off the bottom of this slide, 2017. So the ISO 2006 was set up before the Cronobacter genus was defined. Some people have used the OMPA, PCR, again, you know, for identification purposes. That was 2006, before this. And so the strain collection would be flawed. It has to have been flawed if you're making PCR back in those days. The RPOB, which is used, that was 2009. That's before these species were properly defined and culture collections put together. The CGCA 2013, that's the time. And of course, research is done like, you know, that actual research done like the year or 2 years before the publication. It can take 6 months to a year for the paper to come out. So, as I say, there's a lot of lag time in the system that these dates are not absolute as such, but they're indicative. And clearly 2006 is before 2007. I mean, absolutely. So, ISO 2006. ISO 2006 was replaced 2017. It is not accepted, that replacement 2017 is not accepted in every country. There's one major country that is still essentially working on ISO 2006. Here is the flow for ISO 2006, and it states here incubate at 44 degrees. Now, we know 6% of strains do not grow, and that includes the type strain. And Jeff Farber published the maximum temperature for the type strain, and it isn't 44 degrees. And that was known before this ISO was put out. It also says confirmation, production of yellow pigment. Select 5 typical colonies. It says here, select 1 yellow colony. And yet we know between 2 to 10% of strains do not produce a pigment. And of course, that varies. If you've got a culture collection, which was based on only yellow strains, then therefore you're going to say 99% of my strains produce a pigment, but that's because the starting point was only yellow strains. If you go to clinical strains, you don't get that bias. So it's a bit woolly as to exactly the percentage because it depends on people's starting point. And then down here you've got confirmation biochemical characterization. This is 2006, and yes, I was one of the people who objected to this ISO method on this basis, but it lasted till 2017. Okay, I'll say no more. What we will say is that 2004 we'd already published the contradictions in the kits that were available. Published this in 2004, Carolyn Iverson and I. She was my PhD student at the time, and you got this Enterobacter Sakazaki by API20E, and it's Cloacae and Enterobacters over here, not Sakazaki. Here in API20E, Pantoe, if you do the ID32E, you get Enterobacter Sakazaki. And this was published 2004, 2 years before the ISO method. Later on, 2016, Emily Jackson, another PhD student, and I, we went through, like there'd been an update to the database, we went through and we published this, and we showed the API20E was 82% accurate and the ID32E was 43% accurate. So that's your phenotyping. So if your strains, okay, so here's a little breakdown of that. Here, the speciation according to DNA sequence. We know absolutely what these strains are. But here we look Vitek, and we see the Cronobacter are given us Cronobacter Sakazaki group, whatever that means. API20E, these Cronobacter are given names like Escherichia, Serratia, Pantoe. Okay, and you can just work through this. And here we are, ID32E, and you can just see how the different phenotyping kits and variants give you a different name to the actual correct one. I've already done a video on the 10-year outbreak of neonatal intensive care unit. This paper we published in 2007, but bear in mind those strains were originally identified and isolated in 1994. So they're based on phenotyping. We went through this group of strains. It's in that video. Please look at it. And in that culture collection, really important because that gave us the initial hints about the meningitic pathovirus ST4. But even then, there was this strain that the baby had died from that had been named as a Sakazaki, and by 16S even, we were able to show it's Enterobacter cloacae. So fortunately, and that is in that paper, that correction. Here's another one where with collaborators in China, went through their culture collection from 2005, 2006, okay, went back through their culture collection of 52 strains, re-identified them in 2018, and we found that the original isolation of those strains using the FDA method, is what they claim in the paper, is what we claim in the paper, sorry, I'm a co-author, were actually E. coli, cloacae, and Cronobacter helveticus. Now, the importance is that you might do a recall based upon the presence of Cronobacter. That is a very financially, important or mega decision. Or you might refuse the import because of the presence of Cronobacter. In actual fact, there are E. coli, cloacae, and helveticus. So having that correct identification is really important at the regulatory level, and it can have very, very important financial decisions. Now here's a one which we did back in 2008 with Stacey Townsend, a postdoc with me. And what we did here, we collected strains that initially we believed were Cronobacter Sakazaki. 7 of them came from a hospital. Some were old strains from Moldova, 1983. Some were from Harry Mouchin's 1988 study. So you imagine how old they were. We reanalyzed them. they were not Cronobacter, they were not Cronobacter sakazaki, they were actually Enterobacter homaceae, and actually it revealed that there had been an outbreak, a neonatal outbreak. So here are the strains, here are the dates of the isolation, here's the identification according to phenotyping, and when you do 16S you find that they are Enterobacter homaceae, and we actually have an outbreak here. So these strains, which were initially by phenotyping Cronobacter sakazaki, and you can see the percentages, some are very low, admittedly some are very low, but some are quite high, 98.4%, and they were Enterobacter homaceae. Now bear in mind that organism is an organism for concern. That was a category 2 for the FAO-WHO risk assessments. So Enterobacter homaceae, or the old PCR method. The old PCR method, for serotyping, you had to first speciate, and your primers were based upon Stoop 2009. You can look at the timeline, and Carter 2013. Again, you can look at the timeline for that. In actual fact, we don't find absolutely every strain of Cronobacter actually has CGCA. We don't know why, but we do have some examples where that gene isn't even found. But those primers are based then. There isn't a scheme for Condimenti anyway, it's only for these 6. But why do you have to speciate first? Well, the reason being is because you get the same serotype in different species. The Teresiensis serotype 01 is the same as Malanaticus 01. The Majensky 01 is the same as Sakazaki 03. The Sakazaki 04 is the same as E. coli 0103. Cronobacter malanaticus, and therefore Cronobacter teresiensis, are the same as the E. coli 0129. And it goes broader than that. These are just some examples. So you have to speciate first, because your PCRs for the serotype, you're going to get a different answer. Okay, so you speciate first, and then you come in with these specific primers, and so you get all these PCR reactions according to what the species was in the beginning. So there are 2 schemes. This is what I call first generation. Work by Sun, Jarvis, 2011. They're what I call first generation serotyping, and by this method you get 6% not determined. By the Sun method you get 14% not determined. And then you got this. The Sun method names Sakazaki 05, Sakazaki 06, and the problem was they did not correctly identify their reference strains. The reference strains here were actually Cronobacter malanaticus, and I'm going to admit it, I was a referee and I said this paper should be rejected at this time because those strains needed to be properly identified. The editor ignored me and published the paper anyway. So, that's out there, and it's rather frustrating. But there we go. Now, let's look at serotyping, continuing a bit more. This is a paper in 2015. And when I looked at this paper, they got Serovar down here, and they got Cronobacter malanaticus 01. The thing is, this Cronobacter malanaticus is sequence type 6. And that is not Cronobacter malanaticus. That is Citrobacter cosseri, invasive neonatal meningitic organism. And it wasn't only found in powdered infant formula environment, it was found in semi-finished product as well. You see this PIF BP. So there's a semi-finished product that contained Citrobacter cosseri. Now, for some reason, the authors have done multilocus sequence typing, but they've believed their PCRs instead with regard to what the species is. in that same paper, they then took strains and they then used a DNA microarray to look at the same strains, and you can see, okay, same strains. They've got 4 strains here. They selected 3 of them, and this time using the DNA microarray, they're called Cronobacter sakazaki, and rather than being Melanoticus 01, they're now serotype 04. So we have a discrepancy. What we have the same is a sequence type, which is sequence type 6. So the Cronobacter result, based on PCR, whether it's a DNA microarray or direct PCRs, have given conflicting results. What is consistent is a sequence type 6, which is Citrobacter cosseri, invasive neonatal meningitic organism. Now, that DNA microarray, you can look it up. This is a paper for it. I did a word search, and the name Citrobacter does not occur in that paper. So if it's awkward, but if you don't include other organisms of interest when you design these things, then you're not going to discover them because you didn't design the opportunity for those organisms. But it is a cause for concern to me that Citrobacter cosseri could be present in the manufacturing plant and maybe, you know, in the semi-finished products, and they're being misidentified as Cronobacter. At least that will be a red flag, that will be alert, but we're missing out on the detail that that is a potentially meningitic organism as well. So looking back at papers, 2011, this one, when we saw this paper, we were a bit puzzled, because when we looked at the symptoms of the babies, it wasn't the typical Cronobacter symptoms. So we're a bit puzzled, and looking at the method, there seemed to be a range. We've got biolog here, we've got RPOB primers, you know, being thrown in. So we contacted the authors, and actually this is an occasion where we did get positive result. The person, main author, came across with the strains, and we reanalyzed them and republished it, and they weren't Cronobacter sakazaki. They were Enterobacter homashii and an Enterobacter spur that still, even though the whole genome doesn't allow us to speciate it, but that's it. And we have matches isolated from the unopened PIF. And that is an issue that we would have been interested in 2004, 2006 at the WHO meetings on risk assessments. And you can look back at the earlier slides on Stacey Townsend's 2008 paper with Enterobacter homashii. And if you don't know about that organism, the bottom line, multiple antibiotic resistance, absolutely multiple antibiotic resistance associated with that organism, and very pathogenic. So coming towards the end, we got this one, 2015. These are just some odds and ends I'm just pulling in, just so that, you know, don't just look at the headlines, look at the abstract, look at why they've named an organism. We've got this interesting one on a genome sequence of a hydrocarbon degrading Cronobacter strain from crude oil. Wow, that's interesting. But when you look at it, you see that 99% similarity with Cronobacter pulveris. Nope. So that Cronobacter pulveris doesn't exist anymore. It's Cranconibacter pulveris. But the detail, you realize it's not Cronobacter. But when you look at that headline, if you just go from headlines, you will believe there are hydrocarbon degrading Cronobacter. But this paper doesn't really describe that. So it's 2015. And you think, well, that's 2015, that's a while ago. Let's bring it up to date. Well, yep, here's 2022 paper, prospecting the plant growth promoting activities of endophytic bacteria Cronobacter spur isolated from these leaves. And when you look at the paper, and this is 2022, you can see they've used the old names. Okay, Cronobacter helveticus, Cronobacter zurichensis, and there's their strain. Hey ho, it's not Cronobacter. That's another Cronobacter popping up there. So please look at the detail on the techniques. So identification, detection, the ISO 2007 is an update on 2006. Biochemical identification, those kits are not in the 2017 method, it's individual tests. I've given you the accuracy of the kits as when we analysed them a few years ago, and that's a peer-reviewed paper. PCR probes, just bear in mind when they were designed, just question whether they were designed such that you could differentiate between Cronobacter, Fanconi-bacter and Cicibacter. Look at the timeline and look at what control organisms were used. Personally, we just go for the FUSE allele sequencing. We have thousands of strains for which we have the FUSE curated, and it's not just Cronobacter, it's Citrobacter, Enterobacter, Fanconi-bacter, Cicibacter. They're all there in the database, so we can easily, within seconds, give a species. Multilocus sequence typing varies from the 7 loci, the old one, to the core genome. Default is almost 2,000 loci. That's just a default. You can alter it for yourself. And I think that's what's been really important, is giving us the neonatal meningitic pathovar. Why? We're not sure. Is it because it's more persistent, or is it because it's more pathogenic? That's still, an open question. We have the necrotized enterocolitis pathovar and the adult ones as well. So why use multilocus sequence typing? Well, it's all listed here. You can read this for yourself. Amazing. I'm staggered that it's lasted this long, because I expected it to need updates, and I think the reason it's not, the only update essentially is if you are doing the old PCRs, is that there's some different primer sets to allow for variation, like GLIN-S, were a bit problematic at times. But honestly, whole genome sequencing, it just, I say, 5 minutes, it's all done. It's curated, it's open access, and I think it's expansive, because when we started, we only had 3 genomes. One was Cronobacter, one was Citrobacter, and one was Enterobacter. So it actually was designed to cover a wide range of organisms. I'm going to, my last example is this one. This one we just published earlier this year. Here's a strain that was from a catheter, and this was a catheter from a COVID patient, and the strain from the catheter tip was identified by phenotyping as Cronobacter. It then went through MALDI-TOF and 16S, and it turned out it was a gram-positive. It was a gram variable. In the textbooks, it's gram-positive. The thing is, if you don't get the right gram stain reaction, you can still get an answer by phenotyping. Okay, DNA sequencing, you don't do that. But with phenotyping, if you don't get the right gram reaction at the beginning, you can go off in the wrong direction. So this organism's a gram-positive organism, it's gram variable, and it was mistaken as Cronobacter sakazaki by phenotyping. So that's my last example as a, please get the gram stain right, or bear in mind gram variable organisms. So as I say, where you're going depends on where you're starting from. Look at your culture collection. What is it based on? Is there an unconscious bias in this database? Have we only got yellow strains for a reason, a historic reason? Have we changed Enterobacter to Cronobacter? Because they're not all the same. List of references, most of them are live, or you can just go here and you can access them. Any issues, updates, please. If I said things out of context, or if they have been corrected in the literature, please tell me, and I'll gladly do some corrections myself, and if necessary, a new video. But hopefully this is helpful to people who are new to the Cronobacter research area, and that will help to clarify some issues. So who am I? Well, you can read this in your own time. And thank you for your attention, and hopefully you'll enjoy the next one. Any suggestions, please let me know. Thank you.